Welcome everyone who's joining us here today. I'm excited to share with you a few things about our life sciences labs and show you some cool things that you can do with the Labdisc All-in-One lab solution. Hannah, is there anything that we need to announce on the way in here? Maybe a promo for next week or anything like that?
In today's session, we're going to be talking about the Labdisc, but next week we're going to be introducing a new Labdisc product that's optimized for more of that K5 level. I know some people have already pre-ordered this, and they'll be shipping out later this month. Other ones, or starting into next month as well, and other people are still learning about it. So most of the experiments, or at least some of the experiments we do today, will be able to be done with this new Labdisc as well. So next week's session will be all about this guy. But for those of you who have a Labdisc currently, this is for you. And I'm going to bow off video until I get to come back in a little later.
Sounds good. Hannah is going to be helping me present today, so she is going to be absolutely essential in a few more minutes. But we're going to show a couple things here on my end until we get to Hannah. So welcome in, everyone. If you registered, you probably already know, but maybe you're tuning into the recording later on. Today, we're going to be talking about a series of life science focused experiments that you can do with the Labdisc. That is our portable little all-in-one science device. It's really handy for a variety of lab applications. And as you'll see today, has a wide range of uses throughout the elementary all the way through the high school curriculum. So right now with me, I have our biochemistry model. There's actually 4 different models of the Labdisc plus the Exploris that's coming soon, as Hannah mentioned just a minute ago. But this biochem model here, I'm using it because it has a pH sensor and it also has a heart rate sensor. And both of those are absolutely integral for life sciences education. So what I'm going to start off with here today, if you can refer to my shared screen, is just talking about heart rate. This is a great way to get students interested in the very core of biology. From a very young age, students need to learn about their body, what is inside, what makes it tick, and start to understand how those organs, those structures all work together and the processes inside their body. You may notice here on my screen, I have a human body kit that I pulled up. This is not using the Labdisc, but it's using a 3D printer. Really get some hands-on time with those 3D models to show what the students ordinarily can't see. So here in my printer, I printed off a human heart. This is about life size for an elementary age student. People say the fist is a good analog. So a fist, you can see mine is quite a bit bigger than this heart. So this would be a child's heart size here. And as you can probably guess, I just got that file right here off our MyStem Kits curriculum. You can see the heart model right there. So I would start by introducing the different structures and parts of the body to an elementary audience. Get those students familiar with the things in their body and let them experience them in ways they haven't before. A lot of students have probably heard their own heartbeat or understood their pulse or something like that at this point, especially those later elementary students, but they may not have measured it. And so a graph is also a really key skill to get students understanding how to read and interpret graphs. And you can tie that into the heartbeat with a really simple experiment. I was actually going to have my son do this experiment, but he decided not to at the last minute. So I'm going to be the tester here of our heart rate sensor. So I'm just going to put that on my earlobe. It will also work on a fingertip, whichever is more convenient for you. And I'm going to set up my Labdisc here to start measuring heart rate. And it usually takes just a few minutes for that to sort of acclimate to my body. And so I'll click back over to my Globulab software here. And this is included with every Labdisc. It's a free download, and it interfaces with the device. I'm going to switch over to my meters here and go to 2 meters. And I'm actually going to customize these. Remember, for an elementary audience, we want to make things as simple as we can. So just to start with, I'm going to choose heart rate here and just a simple readout. Then I'm going to go over to this one, and there's actually another option you can do with heart rate. You can choose pulse. And as anyone who's seen a pulse before knows, it goes up and down over and over. So I've chosen one that rises and falls over time. Looks like we have a question in the chat real quick here. Oh, it's from Hannah. Link to download GlobiLab. Thanks, Hannah. That is perfect. Good resource to have. This platform will run on basically anything. It will run on many Chromebooks. It will run on Android, iOS, and Windows and Macintosh computers. So you can see here, I've got my pulse going up and down, as well as an averaged heart rate over time. For those more advanced students, after they get used to something like this, you may want to introduce a more traditional pulse graph. It can be a little bit more complicated for some students to understand, but it's definitely worth taking a look at. So I'm going to switch back to my coordinate plane view here and create a little line graph. I'm going to make sure that pulse is set up on my sensors. Then I'm just going to run my experiment here. And because I've restarted the measurement, it is going to take just a few minutes to adjust to my body, and you'll start seeing that regular pulse come through now. There you go. Pulse rate's going. And you can adjust this experiment to teach students about the role of the heart. You can have students sit quietly and wait, maybe in a nice quiet area of the classroom, read a book for a while, test their heart rate. Or you can have students get up and run around a little bit, start breathing heavily, and you should see that pulse rate increase over time as well. You can do this with the probe attached to them on their fingertip or ear, or you can also pause the experiment and resume later and see how that rate has changed since the last measurement. So heart rate is a very classic experiment for learning about life science for those earlier students. Once they are getting a little bit more familiar with things like graphing and interpreting these graphs, as well as the more complex areas of the life sciences, we'll move up to more of a middle school appropriate lesson. It looks like I have a quick chat message here as well. Yes, thank you, Hannah. If your Labdisc does not come with a heart rate probe, it is available as an extra add-on for other models. The reason I'm choosing to use my biochem model today is because the heart rate sensor is included in the box with that model, but some Labdiscs are not designed for measuring biochemical or really life sciences type measurements. For example, we have our physio model, which is much more for the physical sciences. So it just depends on the Labdisc that you have and whether you've purchased that external add-on. But for many models where you would be doing this kind of experiment, it would come with the device itself. So I'm going to unplug my heart rate sensor here, and I'm going to adjust my Labdisc for my next experiment. So if I pause my screen share here, and you can see on my webcam view, hopefully that's large enough for most of you to see, or you can click on my video to make it a little bit larger. You'll see I actually have 3 cups on the desk in front of me. These are just standard styrofoam cups, but inside each of them is a soil sample or water. So we're going to be expanding now with our middle school lesson to the concept of the environment. What conditions promote what types of animals and plants to grow in certain locations? And one of the main things that we can measure, especially with our Labdisc, is the property of pH. We can test other things with our Labdisc at the same time, like, for example, light, temperature, humidity, all very important things for plant growth, but we can also take a little sample back with us from the exterior of our school. Say, maybe we have a garden plot or at least a lawn in front of our school where we can take a little sample. So I'm going to resume my screen share here so you can see what I'm looking at. And I'm going to show you the lesson that I'm coming from and then walk you through what I was doing. So we have a soil types and pH lesson on my STEM kits. This is the teacher pamphlet here, and you can see all of the background information that I used in order to produce this experiment. And then we also have our student data sheet here. So that's ready-made for you if you would like to do an experiment similar to this one. Now, in order to measure the pH, we actually have to take the soil out of the ground and make it into a liquid slurry, something that our pH probe can measure. If you've used a pH probe before, you can see one on my screen here on my webcam. It's usually a probe that is immersed in liquid in order to measure the acidity or basic properties of that liquid. And so we take that soil, we put it into our cup. We can even use 3D printed sieves. You can see the ones that I used right here on my webcam, printed with various diameter holes to filter out big chunks of earth, sticks, maybe even some insects that have been in the soil or worms. You don't want to get those in your pH sample. But in order to filter out and process that sample with nice fine grain particle sizes, you can use 3D printed sieves or a mesh of some kind that you might have. And then once we process our sample, we add some water and let it sit. So we stir it up, let those chemicals combine into the water from the soil, and then let the soil particles fall to the bottom. So we're not just sticking our probe into sort of grit that will be a little bit harder to read. I also pulled up some images of the areas in my yard where I took these samples. So I'm sure most of you will be able to find something similar near your school. This is nothing fancy from my yard. This is an area that grass has historically just refused to grow. This is difficult soil for grass to get a foothold in. It's very clay based. So if you know anything about soils, that means it's very dense and it's very hard for roots to take hold. So in addition to the fact that it's quite dry in the sun here, when I took this picture at midday, it's also very difficult for roots to get into that soil. So following the procedure from My STEM Kits guide, I dug a hole about 4 inches deep, and I took a soil sample from the bottom of that hole, put it in here. That is sample A on my desk here. Then I did the same thing from one of my garden boxes. I had an area that I hadn't planted anything this year. So this is garden soil. It is a little bit of topsoil and a little bit of potting soil mixed together over the years as I've added new things in there. So it should be fairly rich, loamy soil, not at all that clay consistency. And you can see here as I dug the hole, it had a lot of fine particles that were very loose, dusty soil. So quite a different consistency there. But I follow the same procedure, and that is sample B. So let's take a look and see what we've got here. And if we can make any conclusions about what kinds of pH is in my yard. So I'm going to switch back over here to my GlobiLab software. And because we're just measuring pH, that doesn't really need a graph. I'm going to switch my meter here to show, instead of heart rate, pH. And here we go. I've got my pH probe in the buffer solution, which is about pH 4 for its storage container. I'm going to take it out and I'm going to put it in my water sample right in the middle here. Now here you can see the pH adjusting upward. It does take a little bit to respond, and my water is not perfectly pH 7. This is just tap water from my home. Ideally, of course, in a science lab, we'd be using distilled water and making sure that our equipment has the proper resources in order to get the result that we want. But you can see it's stabilizing pretty close to 7. I'm happy with that. So let's try sample A. I'm going to move it over here. Again, this is the clay soil. You can see, I'll switch it around a little bit and let it adjust over time. When I tried this earlier this morning, I was kind of surprised to see that it was fairly neutral soil. So it doesn't seem that there is a really strong acid or base component to that part of my yard. The reason the grass is growing there probably isn't related to the pH. This is in line with a fairly dry climate where I am in Colorado. That's pretty typical for the soils here to be ever so slightly basic. And then let's swish through my water to rinse my probe. and we're going to go try the other soil sample here, B. This is the loamy soil for my garden. And you can see actually the pH is higher. I was surprised when I tested this. I thought it would be closer to neutral or maybe even slightly acidic in my garden sample, but that is not the case. It actually is slightly more basic than my topsoil in my yard. So it appears that for whatever case, I have a contrast between the loam soil and the clay soil, but it doesn't seem like pH is a major factor in my plant growth there. So I don't necessarily have to worry about that aspect of my yard. But that is something that you could recreate quite easily with pretty much any locality around you. As long as you have soil available, you should be able to replicate that experiment. And you can tie in the environmental constraints for why different animals and plants live in certain areas. It's a very important standard for understanding not only the structure of an ecosystem, but also the processes that go into that, the nutrient cycling, adding things to the soil, plants using those nutrients to grow. That's all wrapped up here, and they can use that to test their very own garden plots. I see some things in the chat here I'm going to check out. So let me go ahead
just
and
me
just
again.
pause for a second here. Okay, Hannah says Kentucky bluegrass does prefer a pH of 6 to 7. Might want to make that soil a bit more acidic. Yes, I have some bluegrass in my yard, but it's not growing in that particular place. It's in a different location that has been a little bit more managed over the years. All right, so that was the elementary and middle school lesson. But I do have one more for you. Hannah has the equipment. This is a dissolved, or not a dissolved oxygen, sorry, a conductivity probe that also in most cases will come with the biochemistry model. I just don't have one available at the moment. So Hannah is going to be my demonstrator here, and I'm going to walk you through the reasons why we would do this experiment, and then I'll let her show you exactly what's going on with that particular experiment. So let me reshare my screen here. And really quick, I'll take you through the rationale behind this experiment. We are going to be looking at a cell membrane. The structure and composition of organelles and parts of the body are very important when it comes to understanding our high school level biology lessons. They're going to need to understand the parts of the body in a lot finer grained detail than they were when it came to the elementary lesson. So in this case, we have a kit available that actually mimics a cell membrane, and you can see Hannah has done a great job designing this, so it actually floats in water. It's very easy to see how the different parts mesh together and naturally form that bilayer membrane for the cell. So if anyone isn't quite familiar with how a cell membrane looks, it has molecules that form a barrier that water can't go in and out, but then it has little channels in it where you can exchange either liquid or also ions, dissolved things like sodium and potassium, nutrients that your body needs. And so we have available a whole series of simulations like this diffusion simulation, where you can show students how chemicals, nutrients might move through a membrane over time. So you can simulate that with computers. Obviously, that's a great visual, so you can see how these particles are mixing. But then once it comes down to understanding why you would do this in a body, probably the best example is a neuron. Your nerves require nutrients like sodium and potassium to be on either side of a membrane, and then these channels to open up in that membrane at the correct time to send that signal to the rest of your body. That's actually how your nerves process information and send signals across your body. So as I'm moving my hand here, my brain is sending electrical signals down my neurons to trigger my muscles. And that all relies on the process of diffusion and transport across a membrane. And we can actually show that in action with the Labdisc. So I'm going to stop my screen share here and turn this over to Hannah as well, where she's going to show you the little setup that we came up with yesterday to demonstrate this.
Yeah, so what I have in front of me here is I just took a simple plastic, Tupperware-esque container, and I took a thin piece of fabric and I hot glued all the way around the edges here, so it's got a really good seal around all the edges. And then I took a nail, and you can see that I poked little channels through the fabric. Now, depending on your fabric, it may already have those built in. But this is what I had around, so I just used some of my scraps. Now I'm going to use the conductivity probe, and I'm going to attach that to my biochem just like you would the pH probe. Slide it on and rotate it right there. And then scroll through until I get conductivity. Now on one side of my barrier, I'm going to put water, and you can see the conductivity of this water is very low. We're going to come in under 1 with basic tap water normally. And then if I move over to my other sample here, it has a much higher conductivity. I actually just used some of the hydroponics nutrients that I did for a recent experiment that I knew made water more conductive. So I'm going to start by pouring the water into one side of my container. And then on the other side, I'm going to pour the conductive liquid. Now my seal wasn't perfect. I could see a little bit of water soaking through, but that's okay. We can just think of that as a, stronger, larger channel in our cell membrane. And so as these two sit here, over time, the liquid from one side is going to start coming into the other side. So you can see that the ions from the conductive side are actually starting to make their way in. Now, the nice thing about this pH nutrient is that it has a color to it as well, so I can visually see as that diffusion is happening. Now, as I move this probe around and try and catch the spots that have some of that diffusion starting to happen, I'm already getting close to 1 down here, and I'm just reading it right off the screen. I could use the GlobiLab software as well, or I can just read it off here. And the longer I leave it, the higher that value is going to be, even without stirring it. But because we're coming up on our, a little longer than our 20 minutes, I wanted to give it a little bit of help to help it along. But this is one way you can demonstrate a cell membrane and track it using your conductivity probe.
Absolutely. You can see in real time the processes that you'll be explaining to your students, that these molecules are going to move from an area of high concentration to low concentration. So they're flooding through those channels into that water. And at the same time, the water is exchanging the other direction as well to equalize across that barrier. The very same process that's going on in our cells right as we speak. All right. Well, thank you for being my demonstration there, Hannah. That looks great. That was put together in just a few minutes, but you can probably put together a more lab-friendly version. I did put in the chat there, and Hannah shared it as well, that there are other common materials just beyond fabric, although the fabric does work really well. Slide transparencies, especially if you get the cellulose acetate version, which are the older version, not Mylar, should actually simulate a cell membrane fairly well, at least from what I've read online. So there are other higher end materials. You can even use something like an onion skin if you want to be very accurate to an actual membrane taken from a living organism. So those are some options for you and some ideas to use this Labdisc portable science lab indoors and outdoors, on the lab bench and out measuring soil pH in the field. So there's a lot of possibilities there. And as you saw throughout, I was referring back to our My STEM Kits curriculum. So if anyone's interested in understanding a little bit more about the lessons and materials we have available for you, just let us know. We'd be happy to talk about that further. But that is our presentation for today. As Hannah mentioned, next week we're coming back. We're learning about a brand new version of the Labdisc. Jillian is going to take us through the Exploris model, which is a lot of fun, especially for those younger students, because it does come with a lot more engagement right on the device with that giant colorful screen there. It's a really great device. So thanks for your time, everyone, and we will see you back next week.